The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone and welcome. Thank you for joining us today's webinar on cell and organ-based culture conditions of the intervertebral disc, which is sponsored by the ORS spine section. My name is Christine Maire and I'm a reader in musculoskeletal diseases at Sheffield Hallam University. I'm the head of the Musculoskeletal and Regenerative Medicine Research Group, which is based in the Biomolecular Research Centre. My key research areas are looking at the cellular biology of intervertebral disc pathogenesis and cellular and biomaterial therapies for intervertebral disc regeneration. I did my PhD at the University of Manchester with Judith Hoyland and Tony Fremont, and I'll be the moderator of today's webinar. I'm an active member of the ORS and spine section, and I currently serve as the secretary of the spine section. The ORS spine section has been created to advance spine research and related sciences so as to improve patient care through basic, translational and clinical research, and to provide leadership representing spine researchers in the Orthopaedic Research Society. And I hope that if you're not already a member, that you'll be willing to join us. Please be aware that this webinar is being recorded and will be available on the ORS website. All webinar participants will be muted during the presentation. If you have any questions, please type them into the questions box in the menu located on the right side of your screen, and we'll address the questions at the end of the presentation. In today's webinar, we have some wonderful speakers. So we've got myself, who I already introduced. We have Professor Fackson Mwale, who is an associate professor at McGill University within orthopedic surgery, and that's based in Montreal, Canada. Dr. Mwale's lab conducts cutting-edge research on tissue engineering of intervertebral disc, and the long-term goal is to promote nucleus propulsus repair using growth factors, scaffolds, and mesenchymal stem cells in the degenerate intervertebral disc. His laboratory is also studying quantitative MRI as a diagnostic tool of intervertebral disc matrix composition and integrity. We have Dr. Davina Permasa, who is an assistant professor in biomedical engineering and Spine Research Institute at The Ohio State University. Her research focus is understanding pathophysiology of disc disease and in vitro and in vivo models of pain disc disease to screen therapeutics. She received her PhD from the University of Manchester with Judith Hoyland and Tony Fremont, and performed postdoctoral work with Dr. James Ioannidis. She serves as an associate member on the ORS Board of Directors and is currently serving on the ORS New Investigator Mentoring Committee, and is also a spine section member and active within the membership committee. Dr. Benjamin Gattenbein is a PhD, is associate professor in biomedical engineering in the tissue and organ mechanobiology group in the Institute for Surgical Technology and Biomechanics at the University of Bern in Switzerland. His research focus is understanding of complex loading to intervertebral discs and ligaments using 3D organ culture, and he's received his PhD in molecular biology and population genetics. He's performed postdoctoral work with both Professor Kito Ito and Mauro Alani at the AO Research Institute in Switzerland, and he's been an active member of the ORS since 2008. So we're going to start off with a presentation with Fackson Mwale, who will start us off by discussing why we might need to culture intervertebral disc cells and tissues in vitro. Over to you, Fackson.
Yeah. Why do we need to culture disc cells in vitro? So this is one of the important things is that it does simplify the system that we are going to study, because there are a lot of confounding factors in the tissue, and so it just makes things very easy if we are going to study a certain mechanism. It also in vitro studies make it possible, for example, to study disc cells from human, because a lot of the responses that we get are often species specific. For example, if you take disc cells from beagles, you find that there are a lot of notochordal cells there. So making extrapolations to the human becomes a little bit of a problem So this is one of the important problems, important things that is in order to study the cells outside the human body. So other advantages is, again, it goes back to what I was saying on the first point, in making a more convenient analysis of if you are going to study disc metabolism, you don't need the whole animal. And nowadays, people are using rabbits, they're using sheep, for example. So you don't need the whole organism to study that. So cell culture, either if you're going to use a tissue culture, or you are going to study the organ, or just a plain cell culture, makes it very convenient to study these processes. Again, the other important thing is that it is amenable, it is cheaper for testing growth factors, drugs, cytokines than the whole organism. So this is quite evident. The other advantage is that it avoids the legal and the moral and ethical problems in animal experimentation. Doing animal studies is good in the sense that you have the whole organism there, but you have to go through a whole bunch of hoops in order to go through the ethical problems, etc. Now, there are disadvantages. One of them is something that I already talked about, is it's quite challenging to extrapolate the results from the in vitro studies back to the intact organism. So this is where in the literature there are a whole lot of problems that we have to address in terms of how we extrapolate the results that we get from in vitro studies. And the other important thing that I'm sure other people on the panel, and Christina are going to talk about, is this problem of de-differentiation. And it's a big problem with disc cells because many of the original cellular mechanisms can be lost, and you start making more fibroblastic cells. So this is a problem that one has to address in terms of it. Now, the other thing, a disadvantage, is that we have to develop standardized techniques in order to maintain the cells healthy and also reproducible for the experiments. So basically what happens is that most people in the lab, in different labs, they feel that they have developed certain standardized techniques, and then we are starting to compare the results from one lab to another, and yet the techniques that they are used are quite very different from what were used in other labs. And this does create a discrepancy, and it's something that we should look out for in both the literature and also in our own experiments. Of course, it takes time to learn aseptic conditions, especially for those who are just starting cell culture. And this is basically it's not limited to disc cells. So this is another problem in the disc. The quantity of material can be limited, especially those taking biopsies. So this has been a big problem in terms of the results that we get in the literature. When you get something from biopsy, you need to expand it. And when you expand the tissue that you get, in most cases, we don't even know what we are getting. And when we expand those cells in culture, the population doublings, etc. And then maybe they are de-differentiated at the time that we start the experiments. So these are some of the disadvantages. In a nutshell, it's great to do cell culture, but one has to look out extrapolations. One has to look out how you expand the small quantity of material that we get. Thank you very much. So Christine.
Thank you very much, Fackson. I will now cover the use of monolayer and 3D culture systems within intervertebral disc cells. So first of all, just a brief summary, and I know many of you listening to the webinar are aware of this because you're very well experienced in the area, but for those that aren't, the intervertebral disc is made up of two main components: the gelatinous nucleus pulposus in the centre and the more fibrous annulus fibrosus around the outside. And within each of these regions there's two different types of cells. So within the outer annulus fibrosus, which is the outer layer around the outside, this is very rich in type 1 collagen, and this contains fibroblastic type cells, and these are termed annulus fibrosus cells, or AF cells. Whereas in the centre, this is rich in proteoglycans, especially aggrecan. So the cells within here produce the aggrecan and also produce collagen type 2, which forms a network within the intervertebral disc. And the cells which reside within the nucleus pulposus are similar but not the same as articular chondrocytes found within articular cartilage. So these two distinct cell types are very interesting to study. So when we want to actually take these out of tissue, if we're lucky enough, we can get a whole disc tissue. So this here is a cadaveric human disc where we can see clearly the gelatinous nucleus pulposus and a clear annulus fibrosus. This one's a nice normal one from a 40-year-old male. So what we first of all can do is isolate out the nucleus pulposus or annulus fibrosus, whichever one we want to get the cells out of, chop it up into little chunks, and then digest the tissue with collagenase most prominently. The collagenase works very effectively because it dissolves or digests all of the type 2 collagen within the actual nucleus pulposus, or type 1 and type 2 collagen in the annulus fibrosus. And this releases the cells from the tissue. These cells can then be strained through a cell strainer and plated into culture flasks where they can be left to settle down. After about 24 hours, we can see cells starting to settle to the base of the flask, and this is just in what we call monolayer culture or adherent population culture. And you can see the cells are just starting to spread out on the actual culture plate. Now, after about 1 week in culture, the cells look more like this. So you can see they look fairly fibroblastic, they're spreading out very rapidly, and they do proliferate fairly fast if the sample is from a normal patient. If the sample's from a more degenerate patient, they are much slower to actually settle down. Now the main problem with this monolayer culture is the cells within there de-differentiate. So this means they lose their normal phenotype, which they experience in vivo. So in vivo, some of the classical things that cells should produce within the nucleus pulposus, as I said, are collagen type 2 and aggrecan. And these at the top here are cells which are directly extracted from disc tissue and then form cytospins, so we can do immunohistochemistry on them. So collagen type 2 and aggrecan are abundant, but collagen type 1 is absent because these are nucleus pulposus cells. However, following culture within monolayer, you can see we lose the collagen type 2, and we lose aggrecan, and we gain collagen type 1 expression, which is not what we want to do if we want to look at what effects cells could have in vivo, because this does not mimic the in vivo situation. The other disadvantage of monolayer culture is that the cells can undergo cellular senescence. So if we look at extended passage of cells, so looking from P5 up to P14, here what you can see is this is telomere length. So if we look here from passage 5 down to passage 14, we can see a clear decrease in telomere length, demonstrating that the telomeres are being shortened and generating replicative senescence. And if we quantify this from non-degenerate MP cells, you can see you get a significant decrease in telomere length over time in culture, which correlates with a decrease in the cumulative population doublings within this culture period. And even more interesting, what these cells actually turn into is they start to become adipogenic. So they start to produce little fat particles. So that's really not what we want if we want to mimic the nucleus pulposus cells. So what we can do is put them into what we call 3D culture systems. And there's a number of cells, such as nucleus pulposus cells. So probably the first system that was developed was pellet, where you basically spin down a high population of cells in a 15 ml Fackson tube. And what you get is these little clumps at the bottom, which over time will increase in size and eventually will look like this. This works on the same principle of mesenchymal condensation during chondrogenic differentiation, which is inducing the phenotype similar to those seen within chondrocytes. Whether this mimics closely to nucleus pulposus cells is not that well understood, but it's likely more to be better suited to articular cartilage. The second culture system, which is also used, is agarose culture, and this is where cells are resuspended in liquid agarose and the agarose is allowed to solidify, and this is then floated about or cultured within culture media. The disadvantage of these two systems is that the cells are very difficult to gain back out of them, which can limit the amount of different analysis you can do following culture. So probably the preferred method for nucleus pulposus culture and annulus fibrosus culture is that of alginate beads. The way alginate bead cultures work is that you resuspend the cells within an alginate solution, which then, when it comes into contact with calcium, causes the alginate to chelate. So alginate is an extract of seaweed, which is perfectly natural and doesn't cause any cell viability problems. And what this happens is when you put it into calcium, you get chelation of the alginate, and cells are then resuspended within the alginate bead, which if you culture them, you can see most of the cells remain single, but you do get some clumps forming. The advantage of the alginate culture system, together with pellets or agarose, is they induce re-differentiation. So cells, after about 2 weeks in culture, start to re-express collagen type 2 and aggrecan, and they lose the expression of collagen type 1. And this ability to re-differentiate is maintained within normal disc cells up to fairly late passage. So passage 9, you can still get good differentiation. So here we have real-time PCR analysis for a number of key genes, so the aggrecan, col2, col1 we've talked about. SOX9 is a transcription factor that controls collagen type 2 expression, amongst other things. So what you can see is in monolayer culture we get downregulation of the normal markers and upregulation of collagen type 1, which is regained back to normal levels within the alginate cultures. However, even in non-degenerate discs, once you passage them too far, they lose the ability to re-differentiate. And in fact, if you take degenerate disc cells, so from a degenerate disc, the number of passages you can take them to before you lose this ability is much lower. And I would recommend never taking them beyond sort of passage 2 or passage 3 before putting them in alginate to allow you to do your studies on in vivo phenotypes. So just to summarise, the monolayer culture is very useful to expand cells, but the cells will then de-differentiate, senesce, and if left too long, can lose the ability to re-differentiate. There's a number of 3D culture systems available which can enable you to regain the in vivo phenotype, and it can be a useful model to look at cellular responses. So I'd now like to pass over to Davina, who's going to discuss the co-culture systems which can be used to mimic the native cellular interactions. Thank you.
Thanks, Christine. So why are co-cultures important? So they allow us to mimic cell interactions in a controlled environment. So why are cell interactions important? They help us to understand and screen regenerative strategies. For example, how MSCs will interact with disc cells and vice versa. And also how disc cells can influence pathophysiology and disease. For example, their interactions with neuronal cells, endothelial cells, and immune cells. So what factors do we need to consider when developing co-culture systems? So we need to look at mechanics, the healthy and diseased states, direct and indirect contact, oxygen levels, media conditions, cell type, 2D versus 3D, and also the time course. So what are the challenges of co-culture? You need to create a balanced system between cell types. What is the optimal co-culture system? So it's really important to maintain viability of both cells, and it's also dependent on the specific question and your output measurements. For example, if your co-culture involves looking at stem cell differentiation, then growth factors and 3D environments are important. And if your co-culture involves neuronal cells and being able to induce neural outgrowth, then you need to be able to actually do this. Also, if you're co-culturing cells in contact, then cell labeling is important too. So you need to think also about what disk microenvironment you want to mimic. So is it the developmental, the healthy mature, or the disease state? So there are a number of different types of co-culture models. These include indirect versus direct contact, insoluble factors, which involve soluble factors versus cell-cell contact. You have 2D versus 3D, which are alginate, pellet, and monolayer cultures, and this is optimal for a particular cell phenotype. You have mechanical microenvironment, and this is important when mimicking native loading conditions in vitro. Also, migration assays, which can be used to examine a cell's ability to migrate in response to another cell. We have explant co-cultures, where the disc cells remain in their native matrix. Also, multi-cell co-cultures, where you actually have more than 3 cell types in one co-culture. Also, there are a diverse array of spinal cell types that have been co-cultured together. And in terms of regeneration, so stem cells have been co-cultured with NP cells, notochordal cells, and annulus fibrosus cells. And also, NP cells have been co-cultured with notochordal cells and notochordal cells with annulus cells. So in terms of degeneration, NP cells have been co-cultured with endothelial and neuronal cells, and notochordal cells with neuronal and macrophages, and AF cells with neuronal and macrophages also. So co-cultures can be performed in indirect or direct contact. So indirect contact involves the secretion of soluble factors that are released between cells. These usually involve trans-well cell culture inserts, and cells are either in monolayer or alginate. So the importance of indirect contact is the fact that you can examine isolated effects of secreted factors and identify mechanism and therapeutic target. So for direct contact, this involves physical cell-cell interactions. So these can be cultured in monolayer, alginate, or biolaminar constructs. And the importance of these cell-cell interactions is the ability to examine cell receptor, cell ligand interactions, the exchange or transfer of cellular components, and also cell fusion. So just going into co-cultures with indirect contact in more detail, these are usually in vitro well plate systems with 2 cell types seeded on the actual insert or underneath, and also in the well plate. They're usually separated by a porous membrane with pores of 0.43 or 8 microns. So co-cultures of human stem cells and healthy human NP cells were performed back in 2006, which involved monolayer cultures of cells in a ratio of 50:50 in normoxic conditions for 7 days. Stem cells have also been co-cultured with degenerate disc cells, as shown here in the figure on the right. So NP cells and AF cells have also been co-cultured with notochordal cells in alginate beads in normoxic and hypoxic conditions for up to 14 days. And here you can see that the disc cells co-cultured in the trans wells with the notochordal cells seeded in the wells. So to examine the effects of healthy and degenerate human disc cells, NP cells have been co-cultured with neuronal cells, as shown here on the right. And neuronal cells were seeded in the well plate so as to measure neurite outgrowth, and the disc cells were seeded in alginate beads in the trans well. So for direct contact studies, monolayer cultures of stem cells labeled with CFDA and NP cells with SNARF carried out, and the formation of gap junctions between the cells assessed, as shown in the figure here. NP cells and stem cells have also been seeded together in pellet cultures, with the NP cells labeled in red fluorescent protein and the stem cells in green fluorescent protein, as shown here. They've also been co-cultured in alginate beads, as shown here. So mimicking the native disc microenvironment is important, as is incorporating cell-cell interactions and the influence of soluble factors. So NP cells and stem cells have been co-cultured with indirect contact and subject to hydrostatic pressure in normoxic conditions for up to 7 days. Stem cells have also been encapsulated by NP cells in bioreactors in hyaluronan gels and subject to physiological loading, hypoxia, and pro-inflammatory cytokines. This is carried out for 14 days. The chemotactic influence of cells in co-culture has been examined using Boyden chambers, which function similar to transwells. Cartilaginous end plate cells have been demonstrated to show an increase in migration in response to increasing numbers of notochordal cells. So whole disc explants from rats, caudal tails, have actually been subject to load and then excised and used to co-culture with macrophages in normoxic conditions for up to 2 days. So co-cultures of more than 2 cell types examine complex cell interactions. So co-cultures of stem cells and NP cells and notochordal cells seeded in alginate beads, and the notochordal cells are labeled in green and the NP cells labeled in orange. And you can see immediately after seeding, all 3 cell types are present, viable. However, after 28 days post-culture, you can actually see that there are no longer any NP cells there. So human AF cells have been co-cultured with macrophages and rabbit notochordal cells in pellet culture, as shown here in the figure below. A number of different spinal cell types have been co-cultured together, which include human AF cells and human neuronal cells in pellet culture. You have human AF cells co-cultured with macrophages, and also NP cells co-cultured with macrophages and cultured with endothelial cells. So just to summarize, co-cultures are essential tools to assess cell interactions in an in vitro setting. The type of co-culture is dependent really on the question that you're hoping to answer, and so whether cell-cell, so direct versus soluble factors, indirect contact are important. And just really thinking about how we can mimic the native disc microenvironment in vitro. So looking at biochemical conditions, so for example, oxygen levels, media that you're using, also, incorporating the loading microenvironment, your healthy versus disease states, and also what cell types are you incorporating into your system. So there are challenges and limitations of the current systems. Not all studies assess cell viability, which is an important parameter when you're optimizing your co-culture conditions. Also, most co-cultures are in normoxia, which doesn't really mimic the native disc microenvironment in vivo, which most cells are exposed to hypoxia. Because of the complex nature of co-cultures, they are underutilized, but they do still remain essential in helping us to answer some fundamental basic science questions related to the intervertebral disc. Thanks.
Thank you very much, Karina. So just like to remind you, if you've got any questions, please do click on the questions box in the menu on the right-hand side of your screen, and we will address those at the end of the webinar. Thank you if you've already posed some questions. So we'll now go on to discuss the use of tissue cultures, which can be done as explants or organ culture systems. So I'm going to start us off with the use of explant culture systems, which is probably the most simple form. So why do we actually want to use tissue cultures at all? What's wrong with just using cells? Well, there is of course a dynamic relationship between the matrix and the cells within our tissues, and therefore in order to understand disc biology, these links between the matrix and cells are essential and important. So the key would be to study the cells as near as the in vivo situation as possible, which of course is within their native matrix within either an explant or an organ culture system, which we'll go on to later in the webinar. So what happens if we take tissue and simply put it into culture without constraining it in any way? Well, what happens is the actual tissue, because it has a high proteoglycan content, will take on the water from the surrounding tissue and lead to massive tissue swelling. This leads to a loss of cellular viability, decrease in cell phenotype, so you get very similar responses to what we saw within the monolayer culture, and you get a decrease in matrix synthesis, particularly collagen type 2 and aggrecan, which of course are essential for the normal phenotype. And interestingly, more recently, Van Dyke has demonstrated that actually putting them into free swelling material can also lead to induction of catabolic responses as well. So there's a number of different explant culture systems which have been developed over the years. And probably the first one was in 1986, which was using the use of polyethylene glycol to increase the osmolarity of the culture media. So this is where the tissue was surrounded by polyethylene glycol and then placed within a dialysis bag, and then the dialysis bag placed within culture media. And this maintained the actual tissue structure, so it prevented the tissue from swelling. And this has been modified more recently by Van Dyke in 2011. Alternatively, there's a number of explant culture systems where the explants can actually be maintained under static load, and then perfusion is used to improve nutrient supply. However, the disadvantage of static loading is it can actually induce cellular death as well. Another system entrapped tissue explants within alginate, so very similar to I described for the entrapment of cells in alginate. This actually entrapped explants into the alginate, which again prevented swelling. And then probably more recently, a couple of different constrained systems have been developed. So one that I developed while I was in Manchester University, where we use a semi-constrained ring culture system, where we maintain an explant culture within a perspex ring that prevents swelling sideways, which actually reduces the swelling vertically as well. And then more recently, an artificial annulus type model by Van Dyke in 2013, which again prevents swelling of the actual tissue and maintains the actual viability and phenotype. So using the semi-constrained system just as a bit of an example, this can be used for human or animal tissues. You can culture non-degenerate or degenerate explants in it, and this has the advantage that often the tissue types that we can obtain from surgery from humans is often fragments of disc tissue. And because this culture system allows us to culture small 0.5 centimetre diameter chunks of tissue, it allows us to even culture those human tissues. So whereas organ culture, which Ben will come to later, you need to have a whole disc present, which often isn't possible with human tissues, this system you can use for degenerate human tissues as well. So if in unconstrained tissue, we look over here, you get a decrease in alcium blue, which is the proteoglycan staining, as compared to our directly extracted tissue, and also a decrease in aggrecan, which of course is a proteoglycan. Whereas in the ring culture system, even up to 6 weeks, the paper's on 3 weeks, but even up to 6 weeks, we maintain alcium blue staining and aggrecan staining. And also importantly, we also maintain collagen type 2, which is the other key matrix component of the actual intervertebral disc. Whereas in the unconstrained tissue, you can see very clearly the collagen type 2 disappears, and in fact you actually get collagen type 1 produced instead. The advantage of these type of systems is also that you can incorporate them in loading. So this is a system developed in Manchester University where you could take the explant culture systems within the perspex rings, put them into little bags and then float these inside a hydrostatically loaded chamber, which allowed loading of dynamic loading regimes and maintained in tissue integrity, cell viability and cellular phenotype up to 6 weeks in culture. So explant systems can be very useful for a number of different applications, and they are very useful particularly if you're looking at degenerate human tissues. So I'd like to pass on now to Fackson, who's going to discuss the use of specialist media to maintain viability and phenotype. Over to you, Fackson.
Thank you very much, Christine. So why do we need specialist media? Basically, when I was looking in the literature about media for the disc, I was shocked to find that there is no specific media actually which is for the disc. There's no specific media even for chondrocytes. So we developed this media that I'm showing here recently, which is called prime growth media, and it is taking into consideration all the things that we know about disc cells But initially, we developed it for organ culture, which is the motion segment with vertebrae. So this media will be coming on the market very soon. It is being it was developed in our lab, and we have licensed it to a company called Wiseend. And so very soon the media will be on the market, and we'll be sending these samples to people who are doing organ cultures. So I want to talk about the media, the media. So the media, this is a living space for cells. And one of the most important things about media is it has to maintain cell phenotype. So if we had, which we define by its matrix production, for the disc cell, it has to produce aggrecan and a high aggrecan and collagen ratio. This is work that we published a couple of years ago. If we had islet cells, the phenotype would be looking at would be that they produce insulin. But in the case of disc cells, my perception is that the most important phenotype is the matrix that they produce. So this is very important to have that kind of media which will be able to produce those kind of increase. The other thing is that you have to look at the collagen 1 to collagen 2 ratio. So this is very important. In the NP, you want to have more collagen 2. In the annulus, you want more collagen 1. So these are the important things that you have to look out in terms of the media. The other important thing is that the media should not impede the scientific objectives of, so it should support the experimental flows. What I mean by that is something that I talked about before, which is basically that if you are going to expand your cells, which you have got from biopsy, your media should not inhibit cell proliferation. So this is very, very important. It should satisfy the requirements of the experimental studies. What is the aim of the study? Is the end product affected by the ingredients of the media? It's very interesting to see when you're reviewing papers and you see that actually, people you are comparing, people have used all sorts of different types of media with all sorts of different types of glucose concentration, and the ingredients are very different, but then they are being, and then you get different results, and then people are wondering why they have different results. So, if again, in terms of in vitro optimization, because you have to optimize the media, the main one, as I said before, is it has to maximize matrix production. For the disc, it has to maximize matrix production. You cannot have enough matrix in the disc. So that is very, very important when you're optimizing this. DMEM was made to keep the cells alive. It was not designed to maximize matrix production, but you'll be surprised how ubiquitous DMEM is in the disc field. So again, I talked about cell division. So cell division, again, this is important. It should not inhibit cell division. Cell division when you are trying to expand biopsies. So these two are important factors in the disc, very important. Now, I come to DMEM, which is Duberger's modified Eagles medium. It contains higher concentrations of amino acids, vitamins, and glucose, etc. But this medium was originally intended for the culture of embryonic mouse cells. They never thought about the disc at all. So this was developed years ago in the 40s. But this is a media that is used for a variety of cells simply because it keeps the cells alive. But we are in a different generation. I think it's important for us to be able to have a media that is specific for disc cells, and even a media that is specific for chondrocytes. So this is very, very important. So another media that is used is RPMI 1640. So again, they are not optimized for disc cells. What we need in a media is these are just some of the ingredients that I'm showing you. It needs to have a higher glucose. The DMEM comes in different flavors, and people in the literature, they just use it interchangeably. They just put DMEM, but it comes in so many different flavors. It has to have an increased sulfate, because if you are going to stimulate protoglycan, you have to have increased sulfate. Most of the students, if you ask, they don't even check what's in the DMEM. Glutamine is important, proline is important, and this is something that we found very recently, that you need a low calcium. Again, DMEM comes, and RPMM, they come in a whole lot of different flavors. So this is very, very important, that in this age, we have to move with the time and have a specific media for disc cells. So this is not the first time there have been media that is specific for certain cells. I'm just showing you here just a few of the media. If you see DMEM, this is here, and this sheet is recommended by pharmaceutical companies what to use. So you see DMEM is actually here. It's for bone marrow and for live cells, etc. But you see there are all sorts of different media. Even macrophages have got their own media. Stem cells, they have their own media that has been developed. Chondrocytes, if you look at chondrocytes, which over the years have been thought to be the cousins of disc cells, look, their media is Hams F12. But if you go in the literature, DMEM is the one that is used. Why? Because of history. You see, DMEM was first used by, when a big person in the field uses it, and it was first used by Dr. Dingo for chondrocytes at Cambridge University. And because Dr. Dingo was using it, so everybody followed. So there's no rationale why DMEM is being used in the disc field. So so this, I'm ending up with a flyer which is coming from the Wisened. So this is called prime growth, and we've got the catalog numbers. And basically, this is just some of the data I'm showing you here. We can be able to culture disc with vertebrae, and you can have a flat surface. This is very important. We've been talking about applying loads, complex loads. You can't apply a complex load without a flat surface, and that flat surface has to come from vertebrae. You can make a flat surface with soft tissues, but you can't apply complex load on that. Ben has been doing a wonderful job of doing applying complex loads, and also Christine. So one of the things that is important is this flat surface. Otherwise, what will happen is that you have uneven loading of the disc. So we have developed this with very easy step. All you have to do is to take the whole motion segment, and you put it in one of the isolation media for just an hour, and you take it out, and it's done. It's ready to be cultured. And as we looked, we also developed a media where you can culture that, which has taken into consideration the history of the disc. What does the disc require? So you can see here, you see if you culture in DMEM for 1 month, you can see this is just cell viability. But for prime growth, we have been able to culture for 5 months, and one of my students has now gone up to 8 months. So you can culture for long, long times, and this is the longest time that disc with vertebrae, with adjacent vertebrae attached, has ever been cultured for more than 6 months. So this media will send it to different labs as samples, to be able to, so that we can be able to do some homogenization of it. So Ben.
Right. Thank you very much, Fackson. So Ben will now discuss the use of organ cultures and the systems used.
Okay, thank you very much. Shall I say good morning or good evening, depending on where you're listening from. I'm going to show you a little bit about the organ cultures and the work we have been doing and other groups have been doing. So, as you have heard, the disc is an organ that is under load, under constant load the whole day, and this load is really varying depending on when we stand up in the morning. We bring the load in the full upright position, and we have really different amount of pressure that is acting on these intervertebral discs. And I think this is a basic understanding we have to have, that there's a low loading, and in some points and during the day we have very high peaks of loading. So this has been shown is really important for these organs. Another important aspect when we compare different species, when we compare a human lumbar disc with a bovine tail disc or an ovine tail disc, so we see here the decrease in size. We'll come back to that also in the bovine tail disc. These are very variable in size, but this has been established now as a very good model system, and I'm trying to explain why this is, and really go and promote that we are going for bovine tail disc as a good model system for intervertebral disc research. Now, when you go down the road here with a rat lumbar disc and a rat tail disc, of course, a rat, and you consider a mouse tail disc work has been published, is nice because you have a lot of antibodies, you have knockout for a mouse model, for instance. But however, when you want to consider physiological studies, nutrition, then there's the Z dimension that comes into play. I mean, you compare now this bovine tail disc, for instance, with this lumbar disc here, you can see that the Z dimension is quite similar. And there's other factors that has been published, as for instance the cell density in these human lumbar discs and compared with bovine tail disc is quite similar. There's a lack of notochordal cells, and there's really numerous studies have shown that notochordal cells are really regenerative type of cells that can influence other cells. So when you do a study, for instance, in a rabbit model, you really have to be aware, or in a porcine model, that this is a complete different animal model and is very regenerative, and is maybe not the closest to the human situation. Now, when we have nutrition for the intervertebral disc, look here on the top left. This is an image with a sagittal view from a human intervertebral disc, and the blood vessel have been stained with a contrast agent. And you can really see here how nicely, how fragile these blood vessels go in capillary system. Go to the cartilaginous end plate, but there is no blood vessel in the healthy disc inside, so whatever. So that means all nutrition has to go through these end plates into the center of the disc. If we look here on the right-hand side, upper right, we see a fluorescent dye laser scanning microscopy image. And this is an ovine tail disc that has been isolated and treated with heparin, which is also glycosaminoglycan, but it's also opening here the blood coagulation, which might also have something to do with this prime growth medium that Fackson Mwale just introduced to you. And what you can see here is when you put it in a free swelling condition in the at Explant molecule, which is a red fluorescent dye, similar size as glucose, you can see that this dye goes really nicely into the center, but it has to go through this pathway here for the inner nucleus pulposus, whereas the outer annulus fibrosus, or this outer layer, are really coming. These are have gained the nutrition from the outside environment. And this below here, this is a study by Benneker and collaborators. They were looking in 2004 in the morphology of these pores, and a number of pores in the cartilaginous end plates. And they have found some patients with increased calcification. So this is another reason that led to the limited nutrition hypothesis, that that when there's calcification happening in these end plates, the nutrition pathways are blocked to these cells in the middle, and maybe this is the reason there is an increased cell death in the center, and we have then maybe failure of tissue by, for instance, mechanical overloading. Going back to culture systems, it has been nicely introduced now that the monolayer is really not a good model for intervertebral disc, but nevertheless, sometimes, depending on the question, you are forced to use the monolayer. You can see monolayer cell-cell interaction, of course, is already very limited for these cells. For disc cells, maybe the 3D dimension is for sure a better system. So here we have hydrogel or tissue explants, as already previously mentioned. And of course, we would aim for organ culture, which is somewhere in between the in vivo animal experiments and the very simplified monolayer. So it has really the microenvironment of the disc can be maintained, and for specific questions, it's very appealing. So we were spending another approach, and how can you prepare a bovine tail model quicker than it has been done previously? So previously we used here, in this step, we use a histological bandsaw with a diamond blade. We cut out these discs from the tail. It's quite tedious. So now we developed a cutter tool where we can use commercially blades that are on the market, and we call it a sushi style cracking off the bone. So the idea behind this is also that we have more open trabecular bones and not grinding them with the diamond blade. And it's also much quicker. And then we use the jet lavage system, and other groups also have been using it, and to open the pores up, and then we have it ready for downstream applications. Now this is a picture of a bovine tail. This is other people listening and also working with that are familiar maybe, but just explain it again. This is a tail. This is more going cranial, and then you have decreasing size of discs. Now for our bioreactor, for instance, that we have built, we're using the middle part here, the mid-sized discs. And be careful when you have these animals from the slaughterhouse. First of all, you need to test that these cells are really alive, that you get the tails fresh enough. And then some animals have still the growth plates present. If you have these growth plates present for the organ culture when after cutting, these can block the nutrition and you will have a decreased cell viability. So we cut it away, make sure that this is cut off properly. And for this, we developed, this is shown here in the mid picture, is shown how you cut these tails. We published a movie and showing all these tools that you, and this is a picture of this very easy and customized cutter tool. So just to point out again, what's really important for us is to keep the disc intact. Of course, it would be a possibility to use whole vertebra and disc, but when you have like these organ culture models, you would like to reduce the number of cells to the cells that you're really interested in, because when the more bone you leave here, the more bone cells you also will compete for nutrition, and maybe this will not work. I mean, when you want to have living cells in the organ culture model. So here, this has to be done manually, and you really have to get a good feeling where to cut. Here, it can happen sometimes when you start doing that you lose some discs because you cut through the cartilaginous end plates. So you should leave maybe ideally 1 to 2 millimeters of this bone here. For us, it was important, as Fackson Mwale already mentioned, to keep a nice and clean surface. This is before the jet lavage system with a ringer solution we use, and then we also add sodium citrate, and the full protocol has been published. And with our system, we cannot grow for months. That's what we know, and this may be a limitation of this approach. But we can show after good cell viability after 14 days, but we can also go to 21 days. But of course, we saw a significant decrease of cell viability. So when you start here is around 18%, 85% in this time, and then here we ended up with around 60, 70%. I just wanted to point out again what also Christine Lemaitre mentioned, is that the adjusting of osmolality is a nice thing to actually diurnally load the disc when you not have a mechanic system. Osmolality will also reproduce this daily shrinkage and expansion of the disc. And the alternative and a new system, as already have been mentioned, is this system by Van Dijk and collaborators, where they use an artificial so-called annulus fibrosus, and they contain these tissue explants by make sure that it cannot swell, and also they have these weights on top of these culture inserts to make sure that this stays immersed in the medium during the culturing. So now going back, what you can do for mechanical loading. So the easiest thing, of course, and it has been done, of course, is to use standard culture plastic to put the disc in standard well plates. This has been done in rapid organ culture model, for instance. Or you can, if you keep these bony surfaces attached and you make sure these discs get enough nutrition, this is a free swelling model where you don't get the full free swelling, of course, because you have the annulus fibrosus fibers that will prevent the full swelling. Maybe you have a 10 to 15% swelling in this situation. A more advanced model requires when you put a force on top. So you will have to design some kind of sinter plates or some contact plates that enable nutrition maintenance in this direction. And what we did actually is to put lead balls with some certain to mimic the diurnal loading and the high loading and the low loading situation, as it's shown here. And of course, as soon as you want to start dynamic loading, you have to think of some containment and to some controlment where you go force controlled or weight controlled. And of course, in this case, you go more force controlled is more important for the disc than for more linear tissues like ligaments. And then you have to think of media refreshment. In our case, for instance, we don't use that anymore because we had a bit difficulties in the past with tubing. Going in and out is, of course, prone for errors. The more tubing you have, the more contamination could happen. And just lately in the literature by the group of Bernice Jim and collaborators, Lisbeth Haglund, Montreal group, they adjusted the shape of the pistons to incorporate the concave shape of the bovine discs. This is a summary slide, what you can actually really do when you have a good model going. So you have here the possibility to put weight, but of course you can then, you can under controlled condition, you can change loading parameters here. You can induce, I call that a catabolic shift experiment. So you inject some proteinases to induce in vitro disc degeneration. Several ones have been proposed. There is the chondroitinase ABC, has been used as an enzyme in the past to treat disc herniation in patients, for instance. Also, trypsin has been come up with, papain has been published, and injection of pro-inflammatory cytokines to induce the catabolic shift. So, for instance, TNF-alpha you can inject. And when you have this disc degeneration established, you can then think of mind experiments to how to regenerate the disc by injection of autologous or allogenic cells like mesenchymal stem cells together or without biomaterials. So it's really, I think it's a really nice playground to study cell therapy experiments, or for instance gene therapy experiments, injection of DNA, or you can think of any strategy basically. So who has dynamic bioreactors currently? So this is just what's collected from the past. So we have groups also in the audience, I think from Montreal, then New York, then we have a bioreactor in Amsterdam, Davos, and Bern, Zurich. And of course, there are more to come, and I've heard also that in Hong Kong they're working on one, and there might be more groups actually, as I've just shown now. So this is the people behind it. I started working with Sibylle Grad and Kate Tomaro Aline in Davos at the AO Research Institute with a bioreactor, and then James Iatridis has a very nice one. We have, I'll just show it in the next slide, with culture human intervertebral discs. Then Theo Smith from Amsterdam, he's using goat intervertebral discs. Then Thomas Steffen and collaborators Lisbeth Haglund, Fackson, Mwale, they use bovine a lot, but also use human intervertebral disc as organ culture models. And then we established one which able to produce 2 degree of freedom loading. This is an overview of these bioreactors. This is the design from Davos. This is a uniaxial compression. Most of them have a peristaltic actuator that is controlled. So it's usually servo controlled servo with air pressure. So it's pulling, and so it's a fluidic muscle. That's the word I was looking for. Sorry. And then we have here this medial flow here. This is the Amsterdam. They have quite a lot of units, I heard, but that can fit into in the incubator. I think 8 units in one incubator. And they have automated media refreshment. And then here also, this is the one from New York. I heard they have force cells that can take a lot of force to culture these human intervertebral discs. And the material is Ultem, which is a material which is very easy to clean and to autoclave. You can see here the design of these two that came out almost the same time. This is now glass, easy to see. Where you have contamination as an advantage. We get rid of all the tubing in our case because we refresh the medium every 2 days. We wanted to make sure we have a good surface for the no slippery effect from the application of torsion. So this is why we are actually building these bioreactors. So why should you come up with a design of a bioreactor? Because we haven't found anything we could take off from the shelf which was able to do a 2 degree of freedom motion. And the disc herniation, I mean, there's a lot of knowledge around that there is an increased risk for disc herniation. And we wanted to study what is mechanical forces that could induce disc herniation. And there's also evidence that when you have increased disc degeneration, there's an increased range of motion in the torsion dimension. And of course, when you design such devices, you have to consider it's not an easy procedure. There is a lot of things you have to consider. Is the bioreactor device? What is the dimension of the incubator? What are you going to deal with? How you control the force? How it's recorded? Do you have backup systems and so on? So this is more the biomedical engineering side of it. This is now how it looks, the final product. We have a 4-unit bioreactor. There's this special serrated titanium plates that can take up the disc if you have a clean surface for the organ from the bovine discs, and then you can apply these torsion movements. This is an example how we apply this. There's other publication available, of course, other examples. This is like a 8 hours high loading, so we call it kind of like a diurnal loading scheme, so we leave it for 16 hours. In this case, with absolute no loading, but compression, 0.2 hertz, and in the other dimension, we had 0.2 hertz slow motion loading and then no loading for 16 hours. This is just how we define this. And then we tested what is a combined compression and torsion, so we overlay both movements and repeated that and left for loading for 8 hours. This is just an example how you could really play with a lot of these mechanical parameters. And actually, when you look at one of these results that we observe is that we found in the center of the disc, in the nucleus pulposus, and on the right-hand side is the anode fibrosis. This is cell viability in percentage from confocal live dead staining. We can reproducibly show that when you apply this compressed twisting motion, that these cells are just dying in the center, but they're not dying elsewhere in the disc. And we try to understand this now by using agent-based finite element modeling and to see why this reduction happens. And I think we found some first clue now. Okay, to summarize what I just talked about now is, so why bioreactors for intervertebral disc research? They're just more complex than the simple monolayer that you have in your lab, and it's more realistic if you want to be clinical relevant. Of course, when you build a bioreactor, this is going to be expensive. This is what I can tell you. But when you have it, compared to in vivo animal models, it's much cheaper. And these organ bioreactors are highly appealing because you can control the mechanics and you can control the culture media, for instance, as Fackson Mwale pointed out. And at the end, you can also, as I already pointed out, you can test translational research. So you can test biomaterials together with cells and whether they're doing any good. Okay, thank you for your attention.
Thank you very much, Ben. So we're now open to group discussion. So we've got a number of questions which have come in already. If anyone has any other questions, then please do put them on the presentation. So the first question I've got is to address to Fackson. Should we consider culturing disc cells in a rash environment similar to what we have in degenerate discs? Otherwise, we should be missing the mark by establishing false expectations of our interventions. So Fackson, first of all, have you got some thoughts on that?
Christine, there's an echo to your. Can you please repeat the questions? There's a.
Jola, can you sort out the feedback? Sorry about this, everyone. Okay, I'll speak a bit slower. So Fackson, should we consider culturing disc cells in a rash environment similar to what we have in a degenerate disc? Otherwise, we could be missing the mark by establishing false expectations of our interventions. Is that clearer?
Yeah, that's an excellent question. Basically, it depends on what your goal is. What's the question for the experiment? If you want to understand how the degenerative conditions work, then you may want to mimic those conditions. So, which means that the pH will be quite low, and then I don't know if you want to have the situation where you have also problems with glucose, etc. But it depends on the question you are asking. I don't think that having a good normal culture media will give you false expectations per se. Basically, what it will give you is the right phenotype for the disc. So you want to start from the right phenotype for the disc. If you are culturing degenerative disc and you have a different question, then you may change the conditions. But I think the starting point would be a situation where you have the right conditions which are giving you the right phenotype for the disc, which is stimulation, which is matrix, increased matrix production, protoglycan and collagen.
Thank you, Fackson. Davina or Ben, do you have anything to add to that question? Davina?
No, I don't have. No.
No. I think Fackson answered it really well.
Thank you. I mean, in my opinion, I've got feedback again. In my opinion, if we're trying to test new therapies, then certainly we need to look at those within a degenerate niche as well. And that's something that we're looking at, is trying to mimic all the different characteristics of the degenerate disc in order to assess new therapeutic applications. So in that kind of question, yes, you certainly do need to mimic the degenerate disc, but as Fackson said, you need to tailor your culture conditions to the question that you're actually answering. So hopefully that's addressed that question. So the next question is, which of the co-culture systems, in your opinion, mimics best the situation in vivo? So I think Davina.
Okay, so which co-culture systems? I guess it comes down to like the question that you're asking, right? So like if you're thinking of like regenerative strategies, then you kind of need to think about like, you know, looking at like effects on phenotype, and kind of looking at really your dependent variables and your output measurements. And likewise with like developing like a co-culture to kind of simulate what's going on in pathophysiology, right? It's like incorporating not just your disc cells, but looking at the extraspinal cells. What other cells are there? So you've got your immune cells, you've got the neurovascular cells also. So really, just to reiterate what like you know, Fackson's comment and your comment about kind of degenerative conditions, I do think it is dependent on the question you're asking. You really need to think about that when you're developing like any culture system. So I don't know if anyone wants to add anything to that. Yeah.
Okay. Anyone else got anything to add there? No? Okay. So the next question is around glucose and oxygen levels. So I suppose Fackson's probably the best one to address this one, as you were talking particularly about glucose levels in media. So there's actually two questions linked to glucose. So one is, in your opinion, what glucose and oxygen levels should be used in 2D and 3D culture? And also there's a question about what's known about glucose levels and how they vary in different species. And should we use different levels if we're culturing cells from different species? So Fackson, do you want to take that one first?
Yeah, so that again is a very interesting question. I was discussing this question with Peter some time ago, and the question was, what are the glucose levels in the human? What are the normal glucose levels in human? Nobody knows the right numbers, but what we know is that they are increased. They are increased, and we know that glucose is the source of energy for nearly every tissue. So you need increased levels of glucose, but in between species, I think there's work that has been done by Gileb and Mark, Risbeth, etc., who have looked at glucose and its effect, etc. So between species, I don't think there's a comparison that has been done as to look at if dog disc has got, but it seems that in the normal disc, in the healthy disc, what you have is increased glucose. And in terms of the oxygen tension in the disc, we know that that the oxygen tension in the disc is quite very different. We also know that related, again, going back to the glucose levels, which also relate to osmolarity. So the osmolarity you are looking for in the disc is something between 350 milliosmoles to about 450. So of course the oxygen tension is low in the disc, and when it goes back to culture, I think it goes back to what Christine was saying, and Divina and Ben, which is basically that that in the disc you have to culture in low oxygen tension. The discs have never seen the amount of oxygen that we give them. It's quite so foreign to them when we culture them. So I don't know if I've answered the question.
Thank you very much, Fackson. So the next couple of questions are around the bioreactors. So if Ben could take these ones. So there's a couple of questions around the use of bioreactors for other animals. So are you aware of any for porcine lumbar discs or murine lumbar discs? That's Ben.
For organ culture?
Yes, for organ culture.
I'm not sure actually. So for murine, well, yes, I think there's a group in Hong Kong. They do murine. I think they have a kind of a loading device for that. I think it should be a group around Keith Luke and I think the Hong Kong group. They were working on such, but I'm not 100% sure. Yeah. Okay. So the porcine, I think maybe someone else can help me up. I'm not sure. I'm not aware of.
Fackson, are you aware of anyone?
No, I'm not aware of. Yeah.
Okay, thank you. Another question.
Thanks.
About the organ cultures. So bovine tail discs haven't got notochordal cells, so they agree that it's more similar to human discs. But what about the motion of the tail and the lack of load compared to humans? So does that cause any problems?
Okay. There has been a study published by Beckstein and collaborators but also Demers and collaborators. They compare the bovine tail, and I think also Fackson Mwale has a publication out where they really summarize what is the similarity from bovine tail discs to the human intervertebral disc. And also Thomas Steffen was pointing out that the shape of the end plate, of course, for when you have apply mechanical loading, is not the same as in a human disc. So it's more a very concave formed end plate. Maybe you remember the slide that I showed from these X-ray images. And this is really something one has to keep in mind. But from the loading parameters, they concluded that it's, when you look at study by Wilke in Ulm, where they loaded ovine tail discs, they also found when you put pressure transducer into this environment that there's a similar pressure as found in a human spine. This is quite amazing. One would guess a tail has not the same pressure on this disc, but this is something like you have to imagine this is a lot of muscle pressure also that keeps these that's acting on these discs and keeps a high pressure as well. So then the question may be for the in vitro, how to what's the best loading parameters? I mean, I think there is no one that can answer you that yet. Good.
Yeah, thank you. So a couple more questions have come in related to the bovine cultures. Just while you're answering that, sorry, Ben. So a question is, so looking at the outcome measures from the organ culture systems, many of the outcome measures are histological outcomes. So are there any other outcomes that you'd recommend that we should look at following organ cultures other than histology? So Ben or Fackson?
So you have to look at protoglycan. Protoglycan in terms of just simple GAG, the contents of protoglycan. I think this is work that's been published. People know how much protoglycan you have in a normal bovine disc. So protoglycan, which can simply be done by GAG assay. Then you have to look at the collagens, the collagen 1 and the collagen 2. And if you feel very ambitious, then you can look at the other proteins and the other molecules in the disc. Because the histology basically will give you, if you do a safranin or stain, that it does stain the protoglycans per se. But of course, predominantly aggrecan. But of course, you also stain the smaller protoglycans such as decorin, biglycan, etc. Because it's simply because of the chondroitin sulfate chains. But you can also so that's another way readout that you can look out quite very simply.
Thank you, Fackson. So I know we're running a little bit over time, but we've got some more fantastic questions. So I think we'll just keep going on for another few minutes answering the questions that have already come in, if that's okay with everyone. So there's another few little questions. So looking at the actual biomechanical forces that we should apply, how can we standardize the biomechanical forces that should be applied onto the disc when in organ culture? So again, probably Ben or Fackson.
Okay, I can take that. So there is a number of approaches, a number of papers published, and we call it simulated physiological loading pattern. So this is like a loading scheme which is of course not bad for the disc. It's kind of beneficial. It's more beneficial than you have like a static loading. Static loading has been shown in various studies. So if you leave it free swelling, is not the best situation. You will have a reduced cell viability. When you put like a little bit of diurnal loading, you make an interchange of high load and low load, you will have an increased cell viability. When you then are in the good situation where you have dynamic loading, you can then play with these, as the good starting point is the simulated physiological, where we had like a very low motion, like 0.2 hertz in the one dimension, and then we changed from high load to low load. We have shown, and the group with Theo Smith has shown that this is good loading parameters.
Thank you, Ben. So a question probably to Davina, a question regarding the conditions required for notochordal versus adult MP cells.
Okay, so for notochordal, I guess some of our previous work has involved culturing them in their native tissue. So based on kind of what they are in vivo, it seems that the notochordal cells kind of like to retain these clusters. However, with MP cells, I think that you can look at seeding them in kind of alginate constructs, because the NP cells in the mature disc are sparse and they don't seem to exist in these cell clusters. So I think that 3D, if you can maintain the notochordal cells in their tissue environment, I feel like that's the best way to culture them to retain phenotype. But you also need to look at kind of biochemical conditions, so like osmolarity, obviously, and other media components that will also pertain to retaining the notochordal phenotype. I hope that answers the question.
Thank you. So I've just got one final question that I'd like to do as the panel. Apologies for the feedback. So when aiming to engineer the nucleus pulposus, is it more important to achieve functional native light matrix composition or an MP like gene phenotype? So does anyone want to take that one?
So this is my perception. So the most important thing in terms of the NP phenotype is what it does. It doesn't matter if it is wearing this jersey or it is wearing these shoes, but it's what it does. And what it does, something we published years ago with human disc, is that for a normal NP, they produce a matrix which has got a GAG to hydroxyproline content of a ratio of about 27:1, which is very different from cartilage. So one thing you don't want is a phenotype for an NP which is basically a chondrocyte. You don't want to put a chondrocyte in the disc because the ratio of GAG to hydroxyproline to collagen for chondrocytes is something like 2:1 or 1:1. So once you go that direction, you are talking about in a degenerate situation. So basically, the most important thing is the matrix that it produces. So in the end, that is what gives function. If you are going to look at in terms of the markers, this is work that has been done by a lot of people in the field. Christine has done a lot of that work. Judith Hoyland has done a lot of that work. And recently, Sibyl did a very comprehensive looking at the phenotype, what it makes, etc., etc. But it's not just one. It's not like a hypertrophic chondrocyte that if you see type 10 collagen and you say, aha, that's a hypertrophic chondrocyte. The NP cell doesn't have that, just one marker that says, here I am. So I think the simplest is to look at the matrix that it produces.
Thank you very much, Fackson. So I'd just like to conclude the webinar. Thank you very much for all your questions. If we haven't managed to address your question during the course of the discussion session, I apologise, but we'll send out the panel's responses to any questions that we haven't managed to cover during the actual session via email following the actual conclusion of the webinar. So thank you very much to all of our wonderful speakers, and thank you to the audience for your active engagement and all the wonderful questions that you provided. Thank you very much.
Thank you very much, and thank you, Christine.
Yeah, thank you. Thanks, everyone.